Oops. So first of all, nucleic acids, if you haven't already gotten to that point, which I hope you have, but this is going to be like one of the first times that I feel like you're going to learn something significantly different than what you learned in bio honors. Because in bio honors, everybody kind of holds DNA and they're like, oh, DNA is the best. It's like it does everything in the cell. And what you're going to learn all throughout the year is that DNA is a very small part of everything. In fact, RNA, which is the other type of nucleic acid, holds a much more important role and is much more versatile in all the things that it does. Okay, so let's talk about what nucleic acids are in the first place. They're a polymer, first of all. So they're polymers made from nucleotide monomers. Okay, so nucleotides, we got to talk about what those are. Nucleotides all have the same basic structure, just like amino acids did. And the basic structure looks like this. Okay, so we've got in the center something called a pentose sugar. Okay, first of all, that's an oxygen on the top. Any spot where there's not a element given, that's carbon. So this is carbon and carbon and carbon and carbon, and then this is oxygen. So pentose. Ose tells us it's sugar. Anything that ends in ose, we know is going to be a sugar. So cellulose, glucose, dextrose, maltose, amylose, fructose, they all end in ose, which tells us that it's a sugar. This is specifically a pentose, so how many carbons are going to be on this? 5 carbons. And so we can expect the basic pentose to have the formula C5H10O5, right? Yes? Everybody agree? All right, great. Coming off this pentose sugar, you're going to have something that is called a nitrogenous base. And I will draw the nitrogenous base as a rectangle or something coming off of this. Now, if you had to guess what a main key feature of the nitrogenous base is, what would you guess? What does it contain? Nitrogen. Yes, you would be correct. So a nitrogenous base contains nitrogen. Good. Then we've got this other thing coming off of here, and that is a phosphate group. I'm going to draw that as like an oval. Okay? And so all together, this thing would be called a nucleotide. Right? But we know that there's not just one nucleotide. There's many different types of nucleotides. So what are the differences? Well, let's start with each of the parts. So the sugar part. The pentose sugar. That's a 5-carbon sugar. Okay, and in RNA, that sugar is going to be named ribose, and it's going to have the formula C5H10O5. Okay, in DNA, it's going to be the sugar deoxyribose. Anybody want to guess the formula for that? C5H10. Then what? Nope. Look at the name. Deoxy. So it's not D-dioxy, it's just single, right? So deoxy means there's just one less oxygen. So this is C5H10O4. Okay? So that's all this means. That's the difference between RNA and DNA is just the sugar, and the only difference between the sugars, what's up? Sure, yeah. The only difference between the sugars is the one oxygen. So you've got ribose that's got C5H10O5, and then deoxyribose that's C5H10O4. Simple, right? All right, let's talk about the differences in the nitrogenous bases. So we can say the nitrogenous bases are the parts that make each nucleotide unique. Okay, so the names of the nitrogenous bases in RNA are uracil. Thank you. Adenine, thymine. Oops, not thymine at all. Sorry, uracil, adenine, guanine, and cytosine. And in DNA, uracil gets replaced with thymine. So you have thymine, adenine, guanine, and cytosine. And so you can see the differences between the two are uracil and thymine. So overall, we could say that there are 5 different nucleotides. The 5 different nucleotides are uracil, thymine, adenine, guanine, and cytosine. Everybody agree? Okay. So, I try not to be disingenuous with you guys in this class. I try to tell it like it is. But there are certain things in biology where it is necessary for me to teach you something that is technically incorrect so that it's not too confusing. Okay? So we're going to learn that there are 5 nucleotides. Okay? Just learn that. 5 nucleotides. That's going to last you at least 4 more biology classes ahead of this one. All right? Now, if you get one more ahead of that, they might tell you about this other nucleotide that occurs in tRNA that sort of interacts with a number of different nucleotide bases. But for now, let's just forget that that exists because it complicates everything significantly. And let's just say that these are the 5 and they're the only 5. Okay? All right. So we've talked about the pentose. We talked about the nitrogenous base. Now we got to talk about the phosphate group. The phosphate group is going to be in all of them. Okay, so all nucleotides contain a phosphate group, but you might see a term used that is a nucleoside, right? So nucleosides are nucleotides without the phosphate group. So you won't see it for DNA or RNA very often, but you might see something like, oh, this thing is a nucleoside complex or something like that. And all that it means is it has the sugar and it has the nitrogenous base, but it doesn't have the phosphate on it. Okay? And I don't want you to see that word and be like, nucleoside, they misspelled nucleotide in the book. It means something a little bit different, no phosphate. Okay? So let's talk about DNA first. DNA stands for deoxyribonucleic acid. Okay? And it's called deoxyribonucleic acid because it's a nucleic acid that has the sugar deoxyribose in it. Right? DNA has one main role, one main job, one main function, and that is it stores the genetic information. Okay? And I've heard some crazy things said about this. I've heard people say the DNA is like the brain of the cell. We don't want to get into that. The brain does a lot of stuff. The DNA is sort of just like a little instruction manual that has like a lot of really good parts in it, and it's got a lot of really like outdated parts that if you followed those instructions, you'd be in a load of trouble. So it's really just all the genetic information that has been accumulated in our genomes or our cells over a really long period of time. There's a lot of stuff in our cells that's in the DNA that never ever gets expressed, right? It's like viruses that have implanted themselves in our DNA that are from, you know, hundreds of thousands of years ago, right? And they just don't get expressed anymore, and they're still there. And so DNA itself is just this like long written out instruction manual, and you got to know what pages to go to. And we'll talk about how we decide what pages in the instruction manual we should go to. Now, it's so important that you don't want it to like get messed up. So normally DNA comes in a double-stranded form. And I would equate this to like the reason why it needs to be double-stranded is not only does it make it really easy to copy it, but also it's like having a backup of your stuff. Right? So when you make a backup, you've got a copy of all of your stuff on your computer somewhere else so that if something goes wrong, you can just restore that. The double-stranded DNA is sort of like that, where you have two copies of it. So if something goes wrong on one side, you've got another side that can sort of rebuild it. Okay? All right. Let's talk about the structure of the DNA, though. Actually, one more thing in this. So we talked about the fact that it stores genetic information, and one of the things that you've heard about in biology probably is somebody has said to you, because they should have said to you, so I'm not saying that they're terrible people for doing this, is that DNA contains the information or the instructions to make proteins, right? Everybody's heard that. It's a thing that you learn. But really, it's technically incorrect because DNA contains only instructions for making RNA. Right? So it's true that certain types of RNA then go on to make proteins, right? But really, the thing that actually gets created from DNA is it's RNA. It's something that's called the central dogma of biology, and the central dogma of biology says that DNA creates RNA, and RNA goes on to create proteins, right? And there's no instance in which DNA directly creates a protein. It always has to go through RNA. So we can say that DNA contains the instructions for making RNA. All right, so let's look at the structure of DNA. First of all, I'm not an artist, so this isn't going to be perfect, but I'll do my best. All right. So this is the double helix, the DNA double helix. We're familiar with this thing. You got these little guys coming out in the middle. Sometimes people call them like the ladder rungs of the double helix. We're just going to rough this thing out, kind of draw it roughly. Something like this. Okay? And so in our drawing here, this part, the blue part, that part is called the sugar phosphate backbone. Okay? And the red part, those are the nitrogenous bases. And you'll notice that the red parts don't ever touch in the middle. They're not solid ladder rungs as people often draw it. And this is good because in order for the DNA to actually do anything, it's got to unravel. And you wouldn't want it to be actually chemically bonded to each other in the middle. So what you want is just an intermolecular force. Now, you don't want it to just randomly unravel. You want something to actually purposely unravel it. So it's got to be a strong intermolecular force. So what is the strongest intermolecular force? Hydrogen bond. Right, so in between all of these nitrogenous bases here, they are going to be bound together, not bonded together, but bound together by hydrogen bonding. Okay? And the hydrogen bonding is always going to occur between specific nucleotides that we call complementary base pairs. And those complementary base pairs are going to be adenine and thymine. We'll complementary base pair with guanine and cytosine, and we'll write that down in just a second. But I want to do some other things first. Okay? So, you'll notice how all of the nitrogenous bases are sticking out towards the middle. Okay? That means that none of the nitrogenous bases are involved in bonds in any way. Okay? So when you form a polymer here, the bonding is going to occur between the sugar and the phosphate. Okay? So in other words, when we go up here, the sugar of one of the nucleotides is going to bond with the phosphate of another one, and then this phosphate here is going to bond with the sugar of another nucleotide. So if you drew this in a very simple way, let's say in our drawing the phosphates will be circles, and the sugars will be pentagons, and then the nitrogenous bases will be squares, like that. Okay? So, in this case, I'm going to draw the unique nitrogenous bases. I'm going to say that this one, let's say, is thymine, right? Then that means that the next nucleotide that comes onto this is going to be right here. Here's the next sugar. And then there's the next nitrogenous base. And let's say this one is, I don't know, guanine or something like that. And then we'll draw one more of these guys. So here's a phosphate group. Here's the sugar. And then let's say this one is adenine. Why not? Okay. Now, interesting thing about this is you can see that the next spot where stuff would bond to this is going to be right here. So we can say this one's moving in this direction. Okay. And when we go down to the opposite strand then, the opposite strand actually goes in the opposite direction. Right? So down here, if we had our phosphate over here, it would be bonded to a sugar this way. And then it would be bonded to a nitrogenous base here. Which nitrogenous base goes to adenine? Thymine does. A to T, yep. And so this one here is the phosphate. Here's the sugar. Here's the nitrogenous base. Which nitrogenous base is that? Cytosine. C, yes. And then this one is phosphate group here. Here's the sugar. Got a little overzealous on my sugar size. And then this thymine is bonded to adenine. Okay? And so they're held in place by hydrogen bonds here. Okay? You can see, kind of obviously at this point, that the sugar phosphate backbone is made out of sugar and phosphate, right? And then the nitrogenous base is in the middle with, and they are made out of, or they're held together with hydrogen bonding. Okay, so just so when we come back we're not confused, these are the phosphate groups. These are the sugars. Anybody want to guess which sugar this is that we've drawn? Which one? It's a pentose, yeah, but which one? Is it deoxyribose or ribose? It's deoxyribose. How do you know? Because you've got DNA and because there's thymine in it, right? So this is the sugar, and it is the pentose sugar. And then the last thing is this, and that was the nitrogenous base. Make sense? So yeah, there's complementary base pairing. And the base pairing works like this. In DNA, the base pairs are guanine base pairs with cytosine and adenine base pairs with thymine. Okay. In RNA, the base pairing goes guanine to cytosine and then adenine to uracil. And there's something called Chargas principle, and Chargas principle says that if we know that they complementary base pair like this, if I know the percentage, so this is my Chargas rules example. Okay, so for instance, if I know that I have, I don't know, 31% guanine in a sample, okay, what other numbers do I know? You'd have 31% cytosine. Anything else? 61%. So that means that there's 61% of the overall strand is guanine and cytosine. Or sorry, 62% of the strand is going to be guanine and cytosine, which is going to leave 38% to be adenine and thymine, right? So if 38% are adenine and thymine, how much is adenine and how much is thymine? 19% adenine and 19% thymine. Make sense? Chargas is the guy that came up with this idea of base pairing, and he did a lot of tests using this principle. Let's talk about RNA. Ribonucleic acid. This stuff is super duper important. Ribonucleic acid has the sugar ribose instead of deoxyribose, and it has uracil instead of thymine in it. But the most important thing to realize is that where we said DNA just stores genetic information, RNA has so many different functions, and so we have different types of RNA that perform different functions. Different types of RNA perform different functions. So you start with mRNA. It's probably the most commonly known type of RNA. mRNA is the type that actually produces proteins. So it takes a copy of the strand of DNA, and then it uses that copy in order to make a protein. It's like the instructions for making a protein. Okay, so mRNA contains a nucleotide sequence that acts as instructions for making a protein. Okay. tRNA is another kind. tRNA is commonly called a transfer RNA, but I think it makes more sense to call it translating RNA because that's really what it does. So tRNA translates the nucleic acid sequence on mRNA into an amino acid sequence. And it painstakingly does that one amino acid at a time. Okay? And if you are making an amino acid sequence, what is an amino acid sequence? What is that? Protein, yeah. So we're making proteins. That's the whole idea here. All right. Now, if you're making amino acid sequence, you got to bond them together. And if you're bonding them together, you've got to have some sort of enzyme that is going to do that because it's going to require energy. So the enzyme that does it is rRNA. We'll say rRNA is the enzymatic RNA that makes up ribosomes and attaches amino acids together. Okay, so it's going to do the job of actually attaching the amino acids together. So some people are like, if proteins have to be made by other things, where did the first thing come from that makes the proteins? Like, where's the machinery, the protein machinery come from that makes proteins if it's required to make a protein? Well, this is your answer. rRNA is the thing. So rRNA, ribosomal RNA, actually makes proteins. It's the ribosome. Yeah.
Can I get the instant?
Sure, absolutely. mRNA, microRNA. This stuff does so much stuff. But the vast majority of the stuff that it does is a regulation. So microRNA regulates cellular processes. So miRNA are small chains of RNA that regulate cellular processes. So what do I mean by this? They regulate things like the cell cycle. And for a long time, we didn't know about microRNA. This is like pretty recently that we've learned about all the things that microRNA does. We used to think there was all this part of the DNA that didn't actually code for anything, and now we're learning, oh, that stuff actually does code for stuff, and it's coding for these microRNA things, part of the stuff that we didn't know what it does, codes for microRNA, and microRNA can do really important things like slow down the cell cycle or speed up the cell cycle. So one of the things that we're figuring out now with cancer is that some cancers are mediated by these very small things of RNA called microRNA, and it can be positive or negative. Sometimes when you have a certain type of microRNA, it's going to cause a cell to, that's being overexpressed, it's going to cause the cell to divide out of control. But the opposite is true where you have other strands of microRNA that suppress the cell cycle. So they figure out when the cell cycle is going out of control, and they actually work to suppress it. So we're learning now that there's all this stuff about the cell cycle regulation that we didn't actually understand, and that it goes deeper than that, and so maybe we can use that as a treatment for people who have cancer. All right, the last thing that rRNA or that RNA does is it is ATP. So RNA nucleosides act as the main energy shuttle for the cell. Okay, so ATP, which is adenosine triphosphate, that is the most common type. Okay, and so it's probably the only type you've ever heard about, ATP. But there are all kinds of what we call nucleoside triphosphates, and the nucleoside triphosphates all do pretty much the exact same thing. They all act as an energy shuttle. So, for instance, we are going to learn in this class that cellular respiration produces roughly 36 to 38 ATP, but it really doesn't. It produces some ATP, but it also produces some GTP, right? Which is guanosine triphosphate. So those are all RNA nucleosides. If you had to guess which RNA nucleotide becomes adenosine, what would you guess? Adenine, right? Because it sounds like adenine. And then if you had to guess which one becomes guanosine, guanine, right? They're just the different bases with 3 phosphate groups on them. And the reason why they're so useful is because they can go through this reaction where you have ATP, which is adenosine triphosphate, and it's going to break down through the process of hydrolysis, and it's going to become ADP plus phosphate plus energy. Right? It's going to yield some energy from this. It's an exergonic reaction, so you can use this breakdown of ATP in order to power reactions in the cell. So once you've got a bunch of ATP, you can use that to power a bunch of stuff. Just like, you know, the sun has energy, but we can't really use the energy from the sun, so we got to like do stuff like charge up batteries, and we can use the energy for batteries in certain things. So all the machinery in the cell uses a battery in the form of ATP, and you've got to convert all the other energy into that ATP form so that all the other processes in the cell can use it. Yeah.
What did you say it was for the ATP to
It's hydrolysis, just hydrolysis reaction, just breaking down things. And so this ADP stands for adenosine diphosphate, whereas ATP is adenosine triphosphate. Questions?